Okay, guys, welcome back to part two of this third module. We have quite a bit in this section of the course. We have genetics, biotech, and then we have some discussion on antibiotics. Now, your book, you will find, has antimicrobial in some of the titles of chapters. We are going to focus on the antibiotics in particular among antimicrobials. We could talk about any number of physical and chemical methods for killing bacteria, and we will talk about them very briefly, but most of this section will be about antibiotics, all right? So this is all about control of growth, right? So it's critical that we understand growth. Our module 2 gave us some understanding as to how organisms grow, what they need to grow, what kind of enzymes do they need to allow them to grow. So these are all related things, right? And now what we're doing is we're applying all of that knowledge, all of the knowledge we've accrued thus far. The structure of these cells in relation to walls and other structures, the metabolism, and the basic needs for growth. So if we need to control microbial growth, and a lot of us in this course are heading into a medical field, so we're looking at most of the time pathogens, which we know are a small percentage, but still we're looking at pathogens and we are concerning ourselves with controlling their growth, right? So there are lots of ways to control growth, from very general to more specific, all right? We could look at physical methods and chemical methods for killing. And we certainly use these all the time, but we should have a basic idea of what physical and chemical means, because when I hear physical, I think of movements, but really physical methods have to do with any number of things, including heat, filtration, radiation, things that some people don't usually connect with the word physical. And then chemical is fairly obvious. There's chemicals, right? There are certain chemicals we can apply to surfaces, whether it be living surfaces on your skin or in surfaces inside your mouth or whatever it is, wherever you're trying to kill, or it could be a surface on your countertop. And when we talk about chemical methods, a lot of times we're talking about disinfecting an inanimate object. So disinfection, using a disinfectant, would be looking at using a chemical to kill on a surface. Usually in this case, when we say disinfection, we're talking about an inanimate surface, not living. Antisepsis would be, on the other hand, applying to surfaces on living things with the intent not to do too much harm to that living thing, and to kill the bacteria only, with those microbes trying to get rid of, okay? Most of this entire section here, this part 2 of module 3, is going to be about the chemotherapy, more specific chemotherapies like antimicrobials, and in particular antibiotics, okay? So we will be focusing on those most of this time, but let's just look at what are some physical and chemical methods before jumping into that. Right? So here we have a chart that's showing you physical and chemical methods, and some of these methods will allow you to actually out and out sterilize a surface. So if we look at the surfaces of glassware or medical instruments we want to use, like dental instruments or some kind of surgical instrument, we can actually put these through an autoclave, right? High pressure oven, we've talked about this before, and basically kill pretty much 100% of any living things there, and even spores, right? We can get up to that high heat and pressure, we can really kill. Lower pressure ovens can do a pretty good job on glassware as well. Or we can boil or flash pasteurize, and in these cases we're working with liquids, and this kills a lot of them, a lot of bacteria and viruses that we want to get rid of. Not spores, but most of the stuff we are concerned with. Again, all physical methods, and you note that they're all heat, right? These top four are all heat. Some will sterilize. Basic definition of sterilization is 100% kill, right? You're getting rid of everything. When it comes to boiling and pasteurization, well, it wouldn't be a sterilization that you have done. You have more or less disinfected an area or a liquid, and you've actually reduced the living things by a lot. We could apply a physical method of filtration with heat-sensitive liquids we can't boil, and this would remove most of the bacteria. And we could also apply some kind of radiation, right, for something that we can't apply heat to, or maybe like plastic instruments and things like that. All right, chemical methods would be any number of these chemicals down here. And these are not by any means the only chemicals that we could talk about, all right? But these are just some really common ones. And you'll notice that some of these say disinfectant next to them because they're typically on inanimate objects, but some of them say antiseptics, skin, antiseptic. So these will remove fewer of the bacteria, but the idea is to do less harm to your subject, right, or your patient, all right? So usually, like with iodine and soap and things like this, these are going to be an antiseptic on a living surface versus a disinfectant when it comes to something like bleach. Now, this is not to say that you can't apply the same chemical as a disinfectant and then turn around and use it as an antiseptic. Usually the key is what concentration of that chemical are you applying. You can apply a high concentration of certain chemicals to an inanimate object and really achieve a higher percent of kill of those microbes, and then you'd have to reduce that percentage when because you don't want to be as harsh on your living specimen. Okay. All right, so let's now jump a little bit ahead and say, okay, we've learned about a lot of these chemical methods, physical methods. Many of these methods we learned really, really early, back when we were, remember Lister, right? Sir Lister in England at the time of Pasteur and Semmelweis and some of these historical figures. He was already applying chemicals to disinfect surgical instruments and stuff like that. But jumping ahead and thinking about more sophisticated ways of killing bacteria, we wanted a magic bullet. We really didn't want to apply a highly concentrated chemical in the body, because that would do harm to our patients, right? So we needed really a magic bullet, and people looked at the craziest things, okay? Well, crazy today, but they didn't know what was exactly the details of it back then. So early in the 1900s, people would use something called salvarsan to treat something like syphilis, okay? So sexually transmitted infection, and salvarsan was used. It so happens that salvarsan was an arsenic compound. That's what it is an arsenic compound. Now, we learned that arsenic, like other heavy metals, like mercury and things like that, are actually non-competitive inhibitors. They inhibit critical enzymes and they're poisons, right? So they would use basically poisons on people to try to treat syphilis. Now, there's lots of problems with this, obviously. It's not a selective way of going about killing because it killed our own cells, right? It was not very selective at all about getting after the bacteria that cause syphilis. There's some horrible side effects, and they take the drug away, and oftentimes syphilis would come back, all right? Later, we ran into some much more successful products like penicillin. Fleming found that penicillin was made by a fungus. In fact, it was poor housekeeping, poor lab keeping, that allowed Fleming to find this really interesting fact, and that is that fungi would produce a chemical to protect themselves, and that chemical would kill other microbes. And this, of course, was used in World War II, and it was ramped up in its production to treat lots of wounded individuals, in war. Okay. Something that's interesting about World War II is, previous wars to World War II, was that a lot of people would die from infections after the war. That a lot of the deaths were not out on the battlefield. A lot of those deaths were a result of injury, and then a surgery was performed, and sometimes not the greatest of conditions, and this would introduce some kind of infection, and people would die from those infections. And of course, there's some other fairly successful work with sulfa drugs, and those were also early 1900s. All right. So what are we looking for with an antimicrobial drug? What do we want in an antibiotic, right? We want a number of different characteristics. One of the characteristics, the key one that we're going for, is I want it to kill bacterial cells and not my own, right? So that's kind of an obvious one. We want selective toxicity. We want to kill the microbe, not ourselves, right? Not the being you're trying to save, okay? We also want a drug that will do what we want it to do to bacteria. Sometimes we want the drug to outright kill bacteria and be done with it. Get rid of those cells. We don't want the infection. But sometimes this can cause real problems. If we kill cells like gram-negative cells, this will release LPS, which is a molecule on that outer membrane, and then our immune system goes haywire and we have shock, we have anaphylaxis. Okay, so sometimes we want a drug that will do just the opposite. It will be microbiostatic. It'll keep them from multiplying, keep the population static, in other words, right? But not outright kill the cells. So it's kind of a strange thing at first to think that you would want to kill them, but this is sometimes what we're going for. So it all depends. The ideal characteristic will depend on what type of organism you're trying to kill. This is one that people always overlook. We want something soluble in water, right? We are water, right? We are mostly made of water. Our tissues are mostly made of water. So we want a drug that will be soluble in water, soluble in our system, so that it can get after those bacteria that are in those tissues. So you'll find that some drugs, some chemicals kill really well in the laboratory, but if they don't dissolve in the water system that we have, then it's not going to be effective, right? We want a long life to the drug. We don't want the drug to break down easily. We need to consider the fact that our body processes foreign chemicals with the intent to destroy them. The liver, the kidneys, these will remove chemicals from circulation, and if it removes the chemical from circulation too early, then it won't be effective against the microbe. We want something that's not allergenic, right? If someone's going to have a major immune response that's life-threatening, not something we want. And this may seem like a trivial thing. If we're going to save lives, then we don't really care about price, but price is a key component, a key aspect that we need to consider, because there has to be some kind of funding behind the production of antibiotics, and if it's too costly, we can't produce enough sometimes of these antibiotics to treat the people that need treating. Okay, so there's a whole host of characteristics we need to keep in mind with making a drug. Not just does it kill something in a laboratory, right? Okay. Where do a lot of these antibiotics come from? Well, there are three major sources for antibiotics. One are molds or fungi, okay? And we already have talked about one of those, and that would be penicillin. We talked about how it is that Fleming identified the chemical coming from penicillium, and that is capable of killing. And so penicillin is the name that it got, right? Because it was from that penicillium organism. Another is cephalosporium, okay? This cephalosporin would be an example from that particular mold. We also find a lot of antibiotics from bacillus species. A lot of bacillus species are found in soil, but not all. And we're going to run into an example of this later. I'm not going to reveal that just yet, but we're going to run into an example of a drug from bacillus species a little bit later on, okay? Actinomycetes. Now this would be a key one. I'd want to jot this down. Actinomycetes are very abundant in soil, and by and large, not all, but a huge majority of our antibiotics come from the soil. And researchers around the world look at soil with the intent to find antibiotics, because it's known that this is a major place that we can find that kind of thing. Okay? So streptomycetes would be an example of an actinomycete organism, and of course streptomycin would be an example. So big one to remember there is that if I'm looking for antibiotics, the soil is a good place to look. In fact, there are researchers that will go all around the world to find unique soil types with the intent to find unique antibiotics. So here we have an example, right? This is a couple years ago, but this happens all the time. People are looking for new antibiotic resistant antibiotics in these soils, and it just so happens that resistance genes also arise because new antibiotics are found in these regions, right? So it kind of one leads to the other when it comes to these resistance genes. And I just add this because, you know, antibiotics are out there in the news, and we can find them. It's all over the place. Of course, right now, most of what you hear about is the virus, right? The coronavirus. But you can go and you can find stuff on antibiotic resistance all over the news. What are some other factors that will influence whether an antibiotic is actually effective, right? We know now that there are certain characteristics we look for in an antibiotic. Selective toxicity, it's got to have a long life, right? You want it to kill or stabilize the population of bacteria. Price, lots of things. What are some other factors that influence whether the antibiotic will even be effective? One is the site of infection. And can we get the drug there? Right? So, in other words, where is it on the human body? Let's say it's human you're trying to treat. Where is it on the human body that this abscess or this infection exists? This influences how we will administer that drug. We need to understand the drug, whether it's capable of getting to that site when we take it orally. If it's not, it will not be effective. Can we make it effective if we inject it underneath the skin? Can we make it effective if we inject it within the muscle, right? These are different aspects we need to consider. The drug's ability to reach the site of infection, and how are we going to administer that? Okay, so these are two major things when it comes to the ability to reach and kill those cells. Another would be the susceptibility of the pathogen we're trying to treat. So in other words, is it resistant or is it susceptible, we would say? Is it resistant to the drug? If it's resistant to the drug and I am able to get it there to that site of the infection, but it's resistant, it's not going to matter, right? So another major thing we need to think about. And also the growth rate of that organism. It may not be susceptible to the drug we're using if the growth rate of the population of cells outpaces the rate at which we can kill the cells. All right? So the overall population will not be susceptible to the drug if the population grows faster than the drug can kill. Right? Okay, and then our last consideration that I want to mention here would be the concentration that you can reach in the body with the antibiotic you're using. In order to be effective, we need to reach a certain concentration of the drug we're using at the site of the infection. So this is a different consideration. It's not a consideration of whether the drug gets there or not. That was our first one, right? Here, this is a consideration of when it gets there, how concentrated is it, and is it concentrated enough to kill enough to affect the population, right? Is it concentrated enough that you can get a handle on that infection? And MIC is minimum inhibitory concentration. We're going to visit it again later, so don't worry. Basically, this is the concentration we need to reach. This will impact the amount of the drug administered to the patient. So let's say I have some kind of infection on my forearm, right? Well, the concentration that the drug will reach here by reaching this location through the bloodstream when I take something orally is going to naturally be less than the dose that I take, because some of the drug that I take won't even be absorbed into the bloodstream. We need to take that into consideration. Now, if we inject this drug, it's possible, depending on the drug type, that it will reach a higher concentration here where my infection is, right? So this will have an impact on the amount of drug administered, the route of administration, speed of uptake is important. We need to understand, okay, if this drug is taken orally or injected under the skin, right, subcutaneously, how quickly will that drug be taken up into the bloodstream? So these are all aspects that we look at in researching these particular drugs. Finally, we have, again, this rate of clearance, this elimination from the body. So this kind of links up with our ideal drug characteristics, right? Long life not only refers to just the chemical nature, how fast does a chemical break down, but long life in the body means we need to consider elimination, how quickly the kidneys and the liver remove the drug, right? So lots of different things that can influence an antibiotic's effectiveness in the body when it comes to killing these bacteria. Okay, so we'll leave it at that for now, and we're going to jump into a bunch of basic terminology we need to be familiar with in this section.